And with that, I'd like to introduce Dr. Khandelwal, who I worked with in Duke, who is going to give you an overview of the complement pathway. And Dr. Khandelwal has very specific instructions to get not too detailed and to keep you all awake during the session, because we know what looking at a complement cascade feels like after a few minutes. So thank you, Sanjay.
Thank you, Dr. Arpali, and I would like to thank organizers for giving me this opportunity to present the complement pathway overview. So I don't have any disclosures to present for today's presentation. So this is my today's presentation outline. So I will be discussing about what is complement and what are the functions of complement and how this complement activation takes place by different pathways, and how this complement activation is regulated, and briefly I will be touching about the complement in pregnancy. So complement system is an evolutionary ancient key component of innate immunity, and it was described early in 1891 by Buchner and colleague as a heat level component of blood that was able to kill the bacteria. And first time they coined a term called annexin. So annexin is a Greek word that means to ward off. And in later 1899, Dr. Paul Ehrlich picked the term complement. So complement consists of about 60 circulating and membrane-bound protein, and these proteins are mainly synthesized in the liver and by the cells involved in the inflammatory response. So now I will be talking about what are the functions of complement, how they are important. So most important function of complement is in host defense, in recognition and clearance of foreign pathogens and antigens. So complement opsonize the foreign pathogens and then induce the phagocytosis of those pathogens, and also do direct killing by the membrane attack complex. And complement also helps in the enhancement of humoral immunity against the pathogen, as well as enhancement of cellular immune response. Complement also has very important function in host homeostasis. This helps in clearance of apoptotic and injured cells and self-antigens. And in pregnancy also, complement is very important because it protects mother and fetus against infection. And since fetus is a foreign tissue for mother, so the complement functions has to be highly regulated for the successful outcome of the pregnancy. So as Dr. Arpali mentioned about the complement pathways, complement can be activated by 3 different pathways. And this complement is a cascade of event where in steady state, these proteins are in inactive form. When there is a trigger, these proteins get activated, and then one protein, once it gets activated, it activates another protein. So the complement activation can happen by 3 pathways based on how this activation is happening, how this is being initiated. And then all these 3 pathways converge at the C3 convertase level, then they make C5 convertase, and ultimately terminal complement activation happens. So now I will go in detail for each pathway. So first, classical pathway. So classical pathway is mainly initiated by the antigen-antibody complexes. And this also can be activated by the C-reactive protein and serum amyloid B. So what happens when these antigen-antibody complexes are formed? First component of complement, that is C1, can bind to these antigen-antibody complexes, and this binding leads to the activation of subunits of C1, that is C1R and C1S. So then they get activated, and once they get activated, they act on the complement component 4, that is C4, and then they cleave it into C4A and C4B. Now, cleaved C4B has binding affinity for complement component C2, and when it binds to C4B, then C1S again can activate it, cleave it into C2A and C2B, and C4B bound to C2A make the classical pathway C3 convertase. Now, this C3 convertase can act on the C3 and then cleave into C3A and C3B, and then ultimately it makes the C5 convertase. Then C5 convertase goes on for the activation of terminal pathway, which I will explain in my later slide. The another pathway is lectin pathway. Lectin pathway is very similar to the classical pathway, but the difference is that how they are initiated. So for lectin pathway, antibody-antigen complexes are not required. But here the recognition happens by the pathogen-associated molecular proteins present on the pathogen. And to detect them, they use phycolins, mono-binding lectin, and collectins. So when this interaction happens, when PAMs bind to these pattern recognition proteins, then similar to C1 complement, mask proteins, that is MBL-associated serine proteins, get activated. And this activation leads to the cleavage of C4 into C4a and C4b. And then similar to the classical pathway, C2 bind to it, and then further cleaves it into C2b and C2a. Then it makes C3 convertase, and similar to classical pathway, again it makes the C5 convertase. So that way, lectin pathway and the classical pathway are similar in their subsequent step, but how they are started is different. Third pathway is alternative pathway. So this alternative pathway is different from both the classical and lectin pathway because it's always active at a very low level in steady state. And the reason being is that it's activated so it can immediately attack on the foreign pathogens. So because of spontaneous hydrolysis, C3 makes C3 water molecule, and then this C3 water molecule binds to factor B. And once it binds to factor B, then in presence of factor D, factor B is cleaved in BA and BB. And that makes alternative pathway initiation C3 convertase. Now this initiation C3 convertase can act on C3, which cleaves C3 in C3b and C3a, and then it again binds to factor B and make the C3 convertase. And complement regulatory protein factor P, that is also called properdin, bind to this C3 convertase and stabilize this C3 convertase. Now this C3 convertase can again cleave the C3 in C3a and C3b, and then ultimately it makes the alternative pathway C5 convertase. So this C3 convertase can again act on C3, and then it can amplify the loop. So that is called alternative pathway amplification loop. And also C3b formed by the lectin and classical pathway can also enter in the amplification loop and can further enhance the complement activation. So now all these three pathways make C5 convertase. Classical pathway and lectin pathway makes C4b, C2a, and C3b C5 convertase, and alternative pathway makes C3b, BB, and C3b C5 convertase. So now these C5 convertase can initiate the terminal pathway of complement activation. So what they do, they act on C5 component of the complement, and then they cleaves it into C5a and C5b. Now C5b, with the help of other complement proteins C6, C7, C8, and C9, makes the membrane attack complex, which inserts lots of C9 molecules in the membrane and creates a pore so that the lysis of the pathogen happens. Now I will be talking about the anaphylotoxin because these are important mediators of the complement function. So anaphylotoxins are low molecular weight cleaved peptides generated by complement activation and induce recruitment of inflammatory cells and inflammation. So C3 is converted by is formed by the cleavage of C3 convertase, and C5a is formed by the C5 convertase. So these two are C3a and C5a are important anaphylotoxins which bind to their respective receptors C3a receptor and C5a receptor. And binding of these anaphylotoxin to these receptors lead to vasodilation and cytokine release, and this induces the recruitment of inflammatory cells and amplify the inflammatory response to kill the pathogens. So these are very important to kill pathogens. Now, so complement is very important against host defense, against bacteria and all the pathogens, but it has to be highly regulated, because if it is uncontrolled, then it can also lead to the destruction of the host cells, which is not good. So all the complement activation pathways are tightly regulated so that they are on whenever they are needed and they don't kill the host cells or host system. So here in this slide, I will be talking about the complement regulatory protein. So there are two kinds of protein. One are soluble protein, another are membrane-bound proteins. So they act differently. So in this slide, the soluble proteins will be shown with the yellow cartoon, and membrane-bound protein will be shown as the pink cartoon. So C1 esterase inhibitor is one of the regulator. It acts on both classical pathway and lectin pathway. In classical pathway, it inhibits the C1R and S. In lectin pathway, it inhibits the function of MBL-associated serine protease proteins. Another complement regulator that is also a soluble complement regulator is a C4b binding protein. So this destabilizes the C3 convertase formed in classical and lectin pathway. Factor H, that is again soluble complement regulator, and it's very important to control the alternative pathway. It destabilizes the C3 convertase, and it also can degrade the C3b formed in the alternative pathway to inhibit the further complement activation. Then there are another proteins called CR1, this is complement receptor 1, and CD55, these are membrane-bound regulators. So they do the decay accelerating factor, so they destabilize the C3 convertase and inhibit the further complement activation. Another complement regulatory proteins are factor I and CD46, that is also known as membrane cofactor protein. So factor I, with the help of other cofactor, it cleaves the C3b and C4 generated by various pathways and cleaves it and inhibit the complement activation. Another complement regulator is, which is present on the membrane of the cells is CD59. So this works at the terminal complement activation pathway and it inhibits the MAC attack complex formation and inhibits the lysis of the cells. To inhibit the anaphylotoxin, there are carboxypeptidase, so they inactivate these anaphylotoxins. And these all regulators are very important because deficiency of these regulators or mutations in these regulators can lead to many pathological conditions. As deficiency of C1 inhibitor lead to hereditary angioedema, deficiency in CD55 and CD59 leads to PNH, and mutations and deficiency in factor H and CD46 lead to atypical HUS. And in HELP also, like there is a dysregulation of complement, which Dr. Bott will talk about in detail about that. And dysregulated complement activation by antiphospholipid syndrome also can lead to the loss of fetus. That will be also talk later in these presentations. So now I will be a little bit touching about the role of complement in the pregnancy, that how complement is involved in the pregnancy and how it is important. So during the pregnancy, immune regulation is very essential for healthy pregnancy because fetal circulation is exposed to maternal tissue and is target of maternal innate and adaptive immunity. And complement are also important to protect mother and fetus from infections. And during pregnancy, there is lots of coagulation activities at the materno-trophoblastic boundaries, which also activate complement. So this suggests that there is an activation of complement during pregnancy, and studies have shown the heightened complement activation during the pregnancy. So this is the study where they studied around 134 pregnant women who were more than 20 weeks of gestational age, and compared the different complement activation fragment into the circulation with the healthy women. So they measure the complement cleaved fragment C3a, C4a, and C5a, and you can see that in each graph on the left-hand side, on the right-hand side, pregnant women's data are shown. So you can see that there was increase in the C3a level, increase C4a level, and increase C5a level. So this tells that there is heightened complement activation during the pregnancy. So to avoid any negative impact of this heightened complement activation, the complement regulatory proteins are enhanced in the embryonic tissue. So decay accelerating factor CD55, membrane cofactor protein CD46, and CD59 are highly expressed in the embryonic tissue, so they can avoid the negative impact of heightened complement activation. So that way, complement is highly regulated during the pregnancy. So here is the summary of my presentation. Complement system is a part of innate immune system which involves around 60 soluble and membrane-bound protein. And this is very important against host defense against infections, and also plays an important role in clearance of apoptotic and injured cells. This complement can be activated by 3 different pathways: classical, lectin, and alternative pathway. And to avoid any negative impact of complement activation, complement is highly regulated by different proteins. And there is heightened complement activation in the circulation during the pregnancy, and to avoid the negative impact of that complement activation, embryonic tissue enhance the complement regulatory proteins.